Hello, everyone. My name is Kelly Berkheimer, and we're going to talk today about how to use a research report to organize your DNA results. So part of what we're talking about today is how to capture that information, how to build some spreadsheets and other ways of capturing things. Also, what is a research report? How can we integrate that into our process? So it's a variety of different topics today, but hopefully by the end, you'll have one or two things or maybe several things that you're interested in trying to see if you can streamline what you're doing for your research. A research report is something that sounds very school-like. We had to write research reports all the time in English class, and you might kind of bristle at the thought. But I'm going to talk today about using a research report in a different way, using it to organize, not using it in a way that brings on anxiety or panic or your hatred of school or any of those kinds of things. But for me, a research report is a place where I can organize my thoughts, I can organize my documents and sources, I can organize my questions and my answers, my hypotheses, my conclusions. So I use it more as a running stream of consciousness. And I'll talk about that a little bit and show you an example. There are lots of benefits to a research report. But I think one of the most important things about it is that you can share it. So you have an organized kind of set of information that you can share. But also, especially with DNA analysis, you can pick up where you left off. So you've researched your matches, you've looked at shared matchings, you've started building trees, you're connecting the dots on who all these people are who are related to you. And then you kind of get to a point where you need more information. You need someone else to test. You need for the testing companies to have a sale and then an influx of people. So a lot of times there's stop and start with DNA research. And so what I do to try to keep organized and not reinvent the wheel is to use a research report format. There are lots of things that a research report can contain. I remember many years ago, I was at a conference, FGS, I believe it was, conference, and my best friend and I, we were trying to decide what session should we go to. And it ended up that it was the end of the day, and there was a session that Judy Russell, the legal genealogist, was doing on writing research reports. And, you know, we both kind of looked at each other like, research reports, well, I don't know. But that was probably one of the most valuable sessions that I have ever gone to in terms of framing in my mind what's the best way to approach a problem, what's the best way to keep track of what you have, what's the best way to form your questions. There are lots of templates online, there are lots of do's and don'ts, there are formal research reports, there are informal ones. Some of them are related to family stories and compiling evidence. Some of them are directly related to a question. And for me, I tend to use the type that has a research question or objective. So I have a new match that shows up in my match list. It's a first cousin. How am I related to this first cousin? And is it a first cousin match? And how do I know what kind of match it is? Those are all the kinds of things that are research questions. Your research question might be, you know, you did a Y-DNA test, but all the folks that are in your Y-DNA haplogroup and surname group are not your surname. What does that mean? How are you related to these folks? Where was the name change? Or maybe there was a misattributed parentage event. So those are the kind of things that we can focus on in our DNA research. Sometimes it's something as simple as, I don't know who this person is, how are we related? So I use research reports for a lot of different questions. Don't be afraid to make it your own. There are a number of webinars, trainings, websites that have research reports, formats, how to use them. I have always found that I'm most successful with something if I figure out how it fits in my normal world. So if you have a particular way that you organize your recipes or your tax documents or your work desktop or whatever it is that you normally do to organize your world, this should be an extension of that, not something brand new and crazy different than what you normally do. But there are 5 F's that I have that can explain and spell failure for you. So one of those is forgetting your own style and workflow. Think about how you normally do things and incorporate your work style and workflow into whatever your research report is. Don't be afraid to not focus on famous genealogists, not look at what does so-and-so do versus what does someone else do. We fall in that trap sometimes of thinking, well, that's a famous genealogist and they do this, and so therefore we're going to do this. It's not necessary. Your research is your research, and make it your own. Some people tend toward formal instead of functional. I am definitely on the functional side. I want that research report to build in efficiency for me. Also, people kind of get tripped up over fancy footnotes and headers and footers and fonts and, you know, all kinds of formatting things. I don't worry too much about that. And I think the first obstacle that spells failure is failing to start. So try this on your next genealogy question or your next DNA question and see if it works for you. See what you like and don't like about it. So what is your goal? Because research reports can help you keep on task. They can help you when projects stop and start so you can remember where you left off. They also help communication with others. So I have had many times where I have been working on something kind of in my own little world. No one's corresponding with me. No one's answering my messages on Ancestry. I'm building trees. I'm looking at the census. I'm solving the problem. And then all of a sudden somebody does want to communicate. So it's nice to have some sort of collection of all of that doesn't take me any time to compile. Also, if you're interested in publishing the NGSQ magazine or something more formal, you know, Research Report can help you also publish for your own family, for your family reunion. So those are the kind of things that you might want to think about. And also preserving findings and documents and sources and stories. I have mentioned before that I've been working on genealogy for a very long time. I started when I was a teenager with my great aunts, and my grandmother's sisters were very into genealogy and going to the places and searching the archives and mailing letters and all the things that happened in the 70s and 80s or even before that. And I was always grateful for their mentorship. And after their passing, I was even more grateful for their written communication, their preservation of what they found. And so my goal as a genealogist and as a genetic genealogist has always been to preserve what I find, to cite sources, to give people a roadmap to get back. So what kind of documents can go into a DNA research report? We're going to talk about a few of those from the different testing companies, how you might want to learn to do screenshots and things like that. So let's just go through the list of some of the testing companies and what kinds of things you might want to have in your DNA research report. So I put a lot of different files along with my research report into folders on my desktop. And on your handout, there's actually a screenshot of what one of my project folders looks like. And so we'll talk about that and I'll show that here at the end. The Excel files that I use are DNA matches from Ancestry, Family Tree, Family Finder, 23andMe. I now have some from MyHeritage. And just over the weekend, Living DNA. My matches started showing up there. I have not yet figured out how to download my matches. Maybe that's a feature that's coming. I hope so. Y-DNA matches, mitochondrial DNA matches, JedMatch matches. Those are all things that I have Excel files of. I have Excel files in JedMatch of all the different tools, the one-to-many or Lazarus project, any number of things. I also have PDF files from reports from tools. I have GEDCOMs from JedMatch. I have screenshots of all kinds of things. So these are the things that either go into your research report or are referred to in your research report. So let's talk for a minute about Excel. Technology is simpler than you think. A lot of people are intimidated by that. You don't have to be an Excel expert to use Excel. There are a couple or three things that once you learn those three things, you can really utilize Excel to a great extent to organize your research. But if you can copy and paste, if you can look for patterns and you can email matches, then you've got this. Don't let it intimidate you. So whether you're using Excel or Numbers for a Mac or Google Sheets, there are classes at libraries, classes at local senior centers, online webinars. And when you don't know how to do something, Google it. So I'll show you what I do when I Google it. We're not going to do a lot of hands-on here, one-to-one, here's how you download. I'm going to show some screenshots of where to find how to download. But your software, if you don't know what to do with it, here's what I do. I go to Google or whatever browser or search engine you use, and I start with how do I? And I do a lot of how do I searches. How do I delete a row in Excel 2010 for a Mac? So I'm asking, how do I do a particular thing in a particular version on a particular type of computer? And then what you'll get generally almost top of the page or page one are videos or a quick explanation or something like that. So if you don't know how to do something, don't let that stop you. I'm going to show you some of the benefits of spreadsheets. And I also have a pre-recorded session at the Southwest Ohio DNA Interest Group in Hamilton County, Ohio. And there's a recording there that has screenshots of how to do these steps, spreadsheets, Chrome browser, how to save PDFs. There are some accompanying handouts that you can email me for. I'm not posting those live on the internet, but if you'd like that, you can email me. And there's a link here to watch that video. Or if you Google Southwest Ohio DNA Interest Group, you can find their page. They have a number of different videos. They started that group maybe 3 years ago now at this point, or a little longer. And there are a lot of great topics on there, from specifics like health reports, DNA health reports, to tools or 23andMe or ethnicity. So there's a great selection of videos there. So definitely stop by and check those out. There's some benefits to Excel. Lots of benefits to Excel, but let me go through the top ones that I use it for. It is searchable. So if I'm looking for someone who's managing groups of kits and I have a downloaded spreadsheet of all my matches at a company, I can search that spreadsheet for an email address. I can search it for a centimorgan value. I can search it for a start or end point of a chromosome match. So all of those things are really important to be able to search for. I also love Excel because of accessibility. I have some vision problems, and so I need sometimes larger fonts. I need colors. I might need to even invert the screen. So there are a number of things that I do for accessibility so that's not so much of a strain on my vision. And also you can see patterns in large amounts of data that you might not be able to see otherwise. Most of the testing companies have a certain number of matches per screen, and then you have to kind of tab your way through the screens to see more and more and more matches. The benefit of a spreadsheet is having all of them in one document where you can see patterns in data. You can see pile-up regions. You can actually visually see that. You can visually see where the start and end positions of a chromosome are the same for a group of people. There are things that you can see that you can't see by tabbing through lists of matches. Also, color coding is really important. I do that quite a bit too, and I'll show you that in a minute. You can download your Ancestry DNA matches to Excel, and I'm going to show you a couple of Excel, well, a couple of Chrome extensions that you can use for that. The first one is one that may not currently work in Ancestry. It was working for a while as long as you didn't opt in to through lines. It worked for a while when you could toggle back and forth and opt in and opt out of through lines. And I believe now it's probably disabled until there's a new version of this. But I wanted to mention it because it's a pretty powerful tool. If you're not familiar with extensions on browsers, almost every browser that I've used has the capability of having extensions built in. They're just little mini tools that can make tasks easier. I'll show you mine in a minute that I use in Chrome. Some of them I use for work, some of them I use for genealogy. But these are the top ones that I recommend that you try. If you're not familiar with Chrome, it is a browser. You can download it for free. It is Google owned, so it is part of the Google universe. And then if you Google Chrome Web Store, what you will find is there are lots of different extensions. And whether you use Chrome or Opera or Safari, you should check in the store for extensions. So the one that I'm going to mention first is Ancestry DNA Helper. And when you download Ancestry DNA Helper into your browser, what installs when you open Ancestry in Chrome are these green boxes on the right. And you can download your matches from Ancestry. So this is a really valuable tool. Like I said, it might currently be disabled, but it might be coming back, so keep an eye on that. And I'll show you what happens. You open Ancestry in the Chrome browser, you click on Download Matches. The scanning can take a long time. You can resume it if it gets interrupted. And what you'll download is a comma-separated value file, which is like a baby Excel file. So you will get a file that downloads to your computer of all of your matches. It'll only work again with through lines turned off. And because I turned through lines on and I'm in the middle of some projects there, I couldn't go in today to check to see if I could toggle off again. There was a time period where you could do that, but I'm not sure that's available now. What you'll get is a spreadsheet, and again, I've redacted some things here. Also, the red type is my notes about who people are. It's not something that came with the spreadsheet, but you'll see here that there's, the name, the administrator of the kit, how many people are in the tree, the relationships. And if you keep on going all the way to the end of this, you'll find that there are live links. So let's say that I want to look at my son Casey and I want to go back to his page on Ancestry. There's a live link that will take me directly back to Casey. So those are the kind of things that are really helpful, is if you can download a spreadsheet of your matches and it includes live links. And I'll show you with the other companies that most of them do that. There are some other Chrome extensions to note. I've highlighted 3 of them here. There are some others, but these are 3 that I've used. Ancestry Match Downloader is a newer one that I hadn't seen in a little bit. Pedigree Thief, I'm actually going to demo that towards the end. And then DNA Match Labeling is one that Blaine Bettinger offers from DNA Central. In case you've ever heard him mention in one of his talks about the dots, the colored dots, there is a Chrome extension for that. So if you want to look at Ancestry DNA Match tool, which is a downloader, there's information in the Chrome store about it. It talks about when it was added to the Chrome store or updated last. And then Pedigree Thief is the same thing. It will create a pedigree chart to pull those names into something that you can copy and paste somewhere else. A lot of people use this tool for Genomate Pro. I do not use Genomate Pro, although I've seen a lot of demos and webinars and things about it. And it's a tool that a lot of people use to do DNA analysis. But I use Pedigree Thief just to capture the names and pertinent data like birth, death, from someone else's tree. And I'll show you on Ancestry how you can do that. I've got just a little quick screenshot here about it, but I can open up a tree that I have, the John Washer tree on Ancestry, and all of these people are long deceased, so it's okay to have all their names and information here. But if you notice at the very top, there is a little snip of what my toolbar looks like in Chrome, what my browser toolbar looks like. And you'll see that there's a couple of three different strands of DNA there that are different DNA applications. One of them is MedBetter DNA. I think one of them is the other two that we've mentioned before. But the one the arrow's pointing to is Pedigree Thief. And what it does is I opened up this tree, I click on Pedigree Thief, and then what shows is a pop-up box that has all of these names in it that can then be copied and pasted into something else. So if you're working with trees and you're trying to figure out who's who, and you want to build a quick and dirty tree, or you want to build a tree for some other family, and you're trying to connect all the dots, this is a great way to capture a lot of information without having to go back and forth between the two trees. You can also see at the very top, if you can see where there is a circle that is half dark, half white, that is actually a Chrome extension for accessibility and for vision. And I use that a lot because it gives me lots of options on changing the vision settings of anything displayed in Chrome. So I just want to point that out because someone asked me about that during the last session. What is that thing that looks like the split circles or split moon? And that's also a Chrome browser extension that gives me the option of changing some of the vision settings. So it's not just invert, although I think invert's a part of it. It also gives me options on how to display things, and it's pretty amazing. So this is what pops up when you use Pedigree Thief, and I'll do a little demo of how that works at the very end. DNA match labeling, again, I mentioned this earlier that this is part of Blaine Bettinger, DNA Central. This is a way that you can add color dot labels to Ancestry. And what you'll see is a screen, I've included a screenshot here from the description in Chrome browser. And you can color code for different levels of grandparents or great-grandparents. And there are 8 different colors that you can use. And so it can help you sort your matches, not just mother versus father, but also a little bit more in depth, another generation or 2 back. And I'm sure that, that there are webinars or little short explanations of these through DNA Central, which is a paid site, but probably also on YouTube and some other locations. So how can you download your family tree DNA matches to Excel? You can download your mitochondrial matches. So if you open up your list and you look at this list and you scroll all the way to the bottom, what you'll see is download matches. And so you have the option of comma separated value file or Excel. You can also make what generates is a spreadsheet of mitochondrial matches. And I have redacted a little bit here, but what you'll see is they're listed from closest to furthest in terms of genetic distance, names, last names, emails, earliest known ancestor, and so on. So there's a lot of great information there. And certainly with mitochondrial DNA, you might want to look at what are the surnames in common, or maybe in column G, you can see that there's in line number 3, Mary Elizabeth Gerhardt, And then in line number 11, there's someone else with Mary Elizabeth Gerhardt listed in the earliest known ancestors for mitochondrial DNA. So you can easily pick that out where you might not be able to view and notice those patterns if you're just tabbing through pages on the tool. You can open your Y-DNA matches and download Y-DNA matches as well. So I've done that for my husband and covered up the, the particulars, but again, you can download that into a file. Now his Y-DNA list is fairly small, so I can look through this and see, I can see Franz Rautenberger, or Rautenberg, 2 different times. That's something I'm going to want to investigate. But, some people have much, much longer lists. And so the spreadsheet is a great way to capture that. But you can also color code the spreadsheet. You can make notes in the spreadsheet. So there's a lot of great uses for the spreadsheet. The spreadsheet of Y-DNA matches looks very much like mitochondrial, but it has the Y haplogroup, the terminal SNP groups, and some of those other things. But it also captures the names, emails, earliest known ancestor, and so on. So that's a really great way to organize. And then as someone new comes in, you can just pop in another row, or you can download the match list again. So from 23andMe, you can download those matches to Excel also. You want to open the DNA Relatives tab, and then scroll down to the bottom where it says Download Aggregate Data. And the nice thing about the 23andMe downloads is that you will get from that segment information. You'll get exact segments you have in common. A lot of that information is captured. And let me just mention particularly about 23andMe, they have a limit to how many matches they allow you to view. And as new matches come in who are closer, higher matches, say higher centimorgan values, the lower matches start to fall off of the list. So another added benefit to the spreadsheet is that you're capturing some of those lower matches that are going to eventually drop off as closer and closer matches are added. So this is one in particular that I go every couple of months and download again, just so that I'm not losing some of those end values. So the spreadsheet for 23andMe, again, very similar to the others. The nice thing about it is chromosome number, start and end position, the number of SNPs tested, and then if you see all the way towards the right, you have links there to compare. You can open up that first one and go immediately back to 23andMe to that match Henry and see all the different things that that you can view about that match. So that's a pretty cool way to do it, to have those live links. Living DNA, as I mentioned earlier, there are matches listed, but they're not done yet in terms of what they're going to offer there. The matches just showed up this last weekend, so mid-May of 2020. They're looking at, I'm sure, all the other different things that they want to offer, but I imagine downloading the match list is going to be one of those fairly soon. You can download MyHeritage matches to Excel, and what you want to do on your match list is in the far right, there are 3 vertical dots. Anytime you see that in any computer program, on any website, you want to click on those dots and see what's hidden there, because there's usually some great stuff. So you can export your entire DNA match list. And so that's where you'll find it on MyHeritage. You'll get an email that your match list is ready when it's ready, and they're going to send it to you through whatever account number you have or account you have there. And if you are administering more than one account, you're going to want to do this for every account. So you're going to want to, if you've got your brother, your sister, your mom, your dad, your cousins on there, and you're administering their accounts, don't forget to export their entire match list too. Remember, we talked about in our clustering last time, you know, that siblings and cousins are really important to giving us depth to our DNA analysis. So make sure you're downloading the match list from all the tests that you manage. MyHeritage will generate a spreadsheet for you that gives you, again, a lot of great information, gives you, you can review the DNA match if you go towards the end there. I didn't expand the columns out, but those are all live links. You can use Excel for GenMatch matches. So there are some instructions on how to do that, and I'm going to leave this here, or you can pause the screen if you're watching. If you want to know how to copy from GenMatch into Excel, there's a couple little tricks to it, but you can follow this. But the nice thing about it is, of course, you can spread out the rows, you can change the fonts, you can change the font sizes, you can color code. There's a lot of great ways to use Excel that make it a lot more visually appealing. The default setting on GenMatch in the one-to-many list is to start with chromosome number 1 and start with the lowest start position. So that's one way that you can use the spreadsheet. Not necessarily the most effective way to start with chromosome number 1, start and end. But there are uses for noticing where chromosomes start and end at the same spot. So there are important things to know there. But I also do a secondary thing where I sort it and filter it by centimorgan matches. So I'll show you that. So what do you notice about the matches here? Looking for patterns again for my son's DNA kit. So this is my son on GenMatch. And what you notice here is that, and these kit numbers are no longer applicable. I've removed all these kits from GenMatch. So I think it's fine to share them because they're not live kits anymore. But what you notice here is that my son's largest matches, most of them right here are John and April, and those are his grandparents. So when you're sorting from largest to smallest centimorgan, you're going to see people there that maybe you've uploaded or you administer, and then eventually you get to the point where you start to see other people who you don't administer their kits or you don't know who they are, and that's where you want to start your research. You can capture GedMatch, GEDCOM searches. So if you're not familiar with GedMatch's GEDCOM tool, this screen may look different now that GedMatch has been purchased by Verigen. I have not been on it since they made that move, but there I'm sure is still a GEDCOM search and a GEDCOM upload tool there because part of analyzing DNA is also analyzing people's trees. So you can search the GedMatch GEDCOMs that are uploaded, and it's a very rudimentary search. It might be a little bit more now that they've had more time with it, but let's say that I'm looking for William Todd, whose father is Samuel and mother is Nancy. I tend not to completely fill this in because it's not a really high-powered search engine that can deal with a lot of fuzziness. So I'm generally just putting in some key names and seeing what comes up, because I want to see if anyone else has linked a tree to GedMatch that includes any of these names. So when I click search, what I find is that there are 4 different kits here that are attached to those names. And they've all been uploaded by one person, one of my cousins. And you can see that they have this data in the tree. And if you want to look at the GEDCOM, you can. And this is sort of what the screen looks like. Again, it might look a little bit different now than it did before. But this is what information is in the tree about William Todd, one of my ancestors, whose father was Samuel, mother was Nancy. He had a wife, Hannah. He had a wife, Susanna. And then these are offspring, their children. So this is something that can be really valuable when you're looking at your DNA matches in GedMatch and then looking at trees as well. And so that would tie you back to 4 different people who have this information linked to them. I sometimes make PDFs of things rather than print. One of the biggest hurdles to organization is having piles. It's also one way to be very inefficient is to have lots and lots of piles of things. And so what I do is I'm not printing out many of the reports like on GenMatch. I'm just making PDFs of them and putting them in the right folder. So I've captured the data, but what I haven't done is I haven't kind of printed and made some kind of, you know, mess that I can't keep on top of. The nice thing about the GenMatch tools is that you will see the kit comparison information. So the top will tell you the tool that you're using and who you're comparing this kit with that kit with names. So that's a great thing for when you go back later and try to remember what it was you did. This is a one-to-one match or one-to-one compare, and it gives you the chromosome start and end locations and segment information for the comparison. So something like this, I'm not going to print. I'm actually going to save as a PDF. And there are a couple of ways that you can do that, but let me show you a couple more examples. One or both of two kits. I use this tool. I used to use this tool a lot when I was doing analysis, the people who match one kit or both kits. And it's really a valuable tool, but a lot of times these reports are several hundred pages. So I'm not going to print several hundred pages, but I am going to save it as a PDF. So in order to save as a PDF, what I do on my PC is I right click and print, or whatever function you use to print. And then I choose either save as PDF or send to OneNote or any number of options, but I'm using the print menu, but not choosing to print. So I save things as PDF and name them. So part of organizing all this stuff involves naming convention, you know, with so many pieces and documents and Word docs and Excel files and all of that, how do you organize that? So I figured out that file naming was probably the most important thing to me. So I wanted to choose something as kind of a general way that I'm capturing information. So I generally am using the tool first. So GenMatch is the tool. Then it's the one-to-one tool or one-to-many tool or one or both of two kits or Lazarus or whatever. And then I'm adding in the names or the kit numbers. And again, your computer is fully searchable. And so using some of those terms, you're going to find the file if you end up losing it. But I set up a folder structure, and I'll show it in a minute, and it's on your handout. But I use a folder structure so that you can see what it is that I do when I'm working on these. So let's look at the sample research report in your handout. But before we do, I want to show you the screenshot of the folder, and this is in your handout too. But what I always start with, I have a folder for each project, and whether that's a project that I'm working on, a collaboration project with a cousin, an unknown parent or grandparent, an adoptee, you know, anything like that, I'm doing the same process over and over and over. And the first document that I have is actually the research report. And it says start here. So that first file there, 00, so it's always on the top of the list, start here, and then I've named the report. So it's usually named by the person or by the question. It might say unknown first cousin for a while until I know who that is. But that's where my running narrative is. That is where I'm noting things to myself. I check this, I check that. And we'll look at that in a minute on the handout. Also, you can see here that I've labeled and numbered these PDFs. First one from GenMatch, second one from GenMatch, third one from GenMatch. And you'll see in the report, it's a redacted report. I apologize for that. I can't share a lot of information, but it will show you that I'm doing the same 3 tests over and over for 3 different people. So the first 3, the 1, 2, 3 files, I'm checking with the client that I have, and Robert. And I'm checking her again on the one-to-one, on the chromosome browser, and on the matching segment matrix. Then I'm checking her against Deborah. And I'm doing one-to-one, chromosome segment matching, matching segment matrix. Then I'm checking her against Brian. So the 789 files are the one-to-one chromosome browser matching segment matrix for Brian. So what you'll see there are the 3 people that I targeted to analyze. And in this DNA report and in this DNA project, this particular person was looking for someone to help jumpstart finding someone. Where do I start? I don't know what to do first. And then what do I do after? So that's why I chose 3 people to target and look at and start to draw conclusions. Then there's also some information below about ancestry matches, JedMatch matches, and then I have some of the other kits. So I just want to kind of let you know that sometimes I number things so that they're not necessarily alphabetized, but they make logical sense, and then I refer to them in my Start Here document. So let's move to how to use a research report. I have my email address here in case you're watching this online and you'd like to see any of the handouts or anything. You can contact me and I will send those to you. And again, this has the recording from Southwest, and it has notes about what we talked about today, but I'm going to scroll down to the Start Here document. So this is just a sample of what I do, either when I'm working with someone who needs assistance or I'm doing this on my own for my own question. And it sometimes starts as a tablet of paper and moves to online. Sometimes it starts online. It varies a lot. Remember, this needs to work for you. So if you're more comfortable in the paper pencil world, do the paper pencil. But I'm just letting you know that this is kind of what my process looks like, and see if you can glean any tips there that will help you. So this was a research report. I was attempting to jumpstart someone to help them find biological father. I have a summary of DNA information. So what do we know? It's always surprising to me how many things I know before I start. And I don't really realize how much I know till I start writing it down. You know, I know this person is my first cousin, and I know that their mother was this and their father was that, and I grew up with them, and I know their kit numbers, and there are all kinds of things that go into a report like this. And locations, if you're working with someone with unknown parent or grandparent, and you're looking for maybe an adoptee or a donor, sperm donor, looking for a parent or grandparents, location makes a big difference. Location can give you clues. So any of those kinds of information I'm including. Then I start with, here's what I did. You can see where it says first. First, I made an Excel file of autosomal matches for so-and-so, and then I insert the file name. Then I made an Excel file of GenMatch matches for this person. Then I analyzed, I sorted this way. Each tab is copied and pasted from GenMatch, and so on. And then I describe what's on each tab of the spreadsheet. I sorted from largest to smallest centimorgan segment match. And here's what I found. I found some matches on chromosome 18, 3, and 6. So remember before when I told you I was looking at 3 different people? Robert, Deborah, and Brian, I think. Gosh, the memory fades. But there were 3 different people I was focused on. So these are their kit numbers and how they have pieces in common with the person who wanted me to help her. So I sorted by chromosome and start position. I'm looking for patterns. I also ran the tool, are your parents related, to make sure that her parents were not related, so we didn't have to worry too much about endogamy or pedigree collapse. Also, I did the same process with the other 2 matches, and I've identified 3 key people to focus on. So you can see where I'm just talking to myself here. I'm just writing down what I'm doing as I'm doing it, so that if I have to set this aside for 6 months and come back, I don't have to reanalyze everything I had and figure out where I left off. So then I ran the one-to-one tool to make sure these people were matches. And these are the 3 that were captured in that folder. I did the people who match one or both of 2 kits. I also have PDFs here that show. In GenMatch, I selected 70 matches and made a PDF of the 2D chromosome browser. So anything that you can print, you can PDF. And so PDFs is probably one of the most stable file forms, and I use it much more than, you know, copying and pasting into Word or anything like that, because I can make a PDF, or I can take a screenshot of the internet and insert it. I also prefer the PDF to a JPEG or a photo format. It's just much easier for me in case I do want to print it later. It's already formatted for page size and that sort of thing. You can tell I also made some PDFs using this matching segment matrix, people who match one or both of 2 kits. And then what I did, and this is the screenshot of the folder when it was finished and ready to hand off, I also included in my research report next steps. What should I be doing next, or what should they be doing next to solve this question? So now it's time to start contacting people. I recommend you contact the match for segment on chromosome 18. If it can be determined that those are mom's side, or you're still not sure, move on to the next high centimorgan match. If it can be determined that those are dad's side, then that information can be used to sort further. And so I'm giving instruction on how to go next level. Then what happens generally with me when I'm working with someone is they do the next few steps. Or maybe the next step is contact this match in JedMatch, or contact this match in Ancestry. Maybe the next step is there really isn't a solution yet. Let's wait until a new match comes in. Let me know. And then the client or you yourself at some other point can come in and just start noting their stream of consciousness and adding to this. So if you're also layering in, let's say you're helping an adoptee and you're looking for unknown parent or grandparent, and you're layering in the census, you're layering in city directories and other things that we use in genealogy to locate people and family groups, those are other things that you can take PDFs of and put them in your research report in your folders. So it's just one way to make one running document, stream of consciousness, and then also make one folder for all the pieces and parts of the project. I'm going to show you the Pedigree Thief because I think that's a pretty cool tool. So let me open Chrome and go to Ancestry. So Ancestry has a lot of different search features, but if you click on search and let's say you search public members trees. Now, we all know the pitfalls of searching public members trees. We know the pitfalls of assuming that they're right. So this is something that I do sometimes to generate some hints. I have the hints turned off on Ancestry so that I don't get hints for other people's trees all the time, because I hardly ever look at them. But in DNA analysis, I might be looking for someone in particular. So I'm just going to use my grandfather's name here. My grandfather's name was Shields Todd. Actually, Shields Franklin Todd. And there's also in my tree Shields Franklin Todd Jr., his son. So I'm just going to search other people's trees for that name and see what shows up. Again, you can put in all these the other information that you know, but oftentimes I find what I need within the first few hits, if it's there at all. So let's say that I search for that and I find that there's a tree here where Shields Franklin Todd is listed. And again, I don't necessarily know which Shields Franklin Todd. It looks like this one is my uncle, and it looks like this one is my grandfather. So the first one, the little bit younger one, is my uncle. The little bit older one is my grandfather. So I'm going to search the Surin family tree. I want to look at that tree. So if I open that tree, there's a lot of information here, and this is what this person has accumulated on my grandparents. Brothers, sisters, you know, all the things that show up there. So what I'm going to do is I'm going to go to the view tree. And how many of you get to this point and you're like, I have no idea what to do here? I can search this tree for my grandfather or my uncle and find the name, but then you're only allowed to expand one of these at a time. So you can only click on one at a time to expand them. It takes forever. So what I want to do is I want to use Pedigree Thief so that I can capture the main pedigree of this tree. So I'm going to go up to the browser, and if you can see it up here in the upper right corner, my mouse is hovering up there where it says Pedigree Tree, or sorry, Pedigree Thief. If I click on that, what happens is the tool starts scanning, and it doesn't take too long. See how it's scanning all the different branches? And then what generates is the document that, or a list that we can copy and paste. So just like that, you can copy this whole thing and you can print it into a Word doc or any of that. You can have dates in full added to it. And if you scroll through here, you'll see these are all the major lines of the tree. It's not every single person in the tree. It's the major lines of the tree. But then if you need further information, you can go back and get it. So this is what I use to capture information from other people's trees, where I'm not necessarily using their tree for research, but I might want to see what they have with regards to a certain name. So I want to thank you today for joining in on this session of Genetics, Genealogy, and You. This is a free group that meets on the third Sundays several times a year, 6 or 7 times a year, not every month, but we do meet whenever there's not a holiday. So our next meeting will be in July, third week of July. We're skipping June because it falls on Father's Day. So if you have any questions or you'd like to get a hold of me, Genetics, Genealogy, and You, I have a Facebook page. You can look for me there. We have an email distribution list that just has a reminder about the meeting, or you can ask any questions. I also send handouts through that. So Genetics, Genealogy, and You. Again, my name is Kelly Bergheimer. And if you're interested in joining or being added to our list, just email and let me know. Thanks for joining today.